what I'm going to be telling you today is on HIV cure, but I'm going to give you examples that have been done in our own lab. So in the last 5 years, well, we've been doing some advances, and most of the examples you're going to be seeing today are things that are been in our laboratory. So to start with, and by the way, and I want to thank the organizers for the invitation, especially Victor, for his kind invitation to be here today with you. So having said that, first of all, I want to give you a flavor of where we are. We are in Badalona. Badalona is nearby Barcelona. I'll show to you, and this is the institute where I do work, IRSI Caixa. IRSI Caixa is a kind of funny experimental story. It's a private, non-for-profit institution, and is partnership between a bank and the government of Catalonia. So it's been running now for about 30 years, 35 years, and it's been very successful. And we start working with HIV and AIDS epidemics, but now we switch in the last maybe 15 years to other emerging diseases. Obviously, we got very deeply into SARS-CoV-2, but there are other viruses that are tackled as well, not only from the virology, but also from the immunology point of view. The second thing is that we are based in this area. We are in the top of a hill. We have Barcelona downtown on the background, the sea, and it's a wonderful landscape to work and to do research. This is the whole campus, and the major building in the campus is obviously the hospital. This is one of the 3 major hospitals in town, and we do follow a large cohort of people with different infectious diseases, including HIV. And IRSI Caixa, this research institute, has a particularity that is not alone. It's not a standalone experiment. It works in collaboration with the hospital. And the reason I brought this slide is that we are close, the laboratory, IRSI Caixa, and the infectious disease department. So we are in the same corridor. We are, you know, when we get samples, it takes basically 2 minutes to get from the blood draw room to the laboratory. The quality of the cells is very good because of that. So we do have many studies in, as I said, immunology and different infectious diseases. We are about 120 clinicians, 120 scientists within the same space. So this is very rewarding, I can tell you. Finally, because I said that most of the things that I will be introducing to you today are from our own lab, this is the team, and most of the things I'm going to be telling you today are from these people in the picture. These people are mainly dedicated to viral immune pathogenesis and viral persistence. So let's go for that. You all know this story. I always want to say that the key points on HIV have been 81, first AIDS diagnosis, 83, HIV discovery here in France, 87, first antiretroviral therapy, ACT, and 96, triple drug therapy. But don't forget ever that the story really started in the 1920s with the interspecific jump from SIV in non-human primates to HIV in humans. And I always, when I give this talk, I always want to remind people that there are the 3 only things you have to remember in basic research in HIV are these 3. So first is that it's a virus that infects the immune system. And that makes this virus different from any other virus. And this is a real problem because it weakens the immune system. The second point is that it's a virus that integrates in our own DNA. And that is different from other viruses as well. Remember, we can cure hepatitis C, but hepatitis C does not integrate in the human DNA. And the third point that is also critical is a virus, it's an RNA virus that diversifies very quickly. And this diversification has drawn us nuts, especially in order to design vaccines. This diversification is what makes that we can hardly cope with the virus in terms of getting neutralizing antibodies to do effective vaccines. So these, I would say, these are the 3 most important things you have to remember about HIV. And this is the point. We have a virus, we have some treatments, we treat people, and this is a cartoon of viral load on the left-hand side and time on the x-axis. And we treat people, viral load gets down to undetectable. So good, so far so good. But we know that despite of that, we can treat HIV, but we cannot cure HIV. And that is because there are reservoirs of virus that stay in the latent reservoir of different CD4 T cells. And if we stop therapy, viral load will come back. And this is the general framework of this presentation. And from here, we'll try to analyze a little deeper what's going on here. So one important thing is that this story about persistence is extremely complicated. I can tell you that in the last 15, 20 years, we've known a huge amount of new things about HIV reservoirs. And those things are basically compartmentalized in three areas. One is the molecular area, which is in the center down of this slide. I don't think the pointer is working anymore. So in this area over here, where we try to understand the interaction between the virus and the human genome, but that happens in too many different cell types, not just one cell type. It's not only a question of CD4 T cells. It's a question of many different cell lines that are in the body and they can be infected by HIV. And at the same time, because lymphocytes circulate through the body, that it will show up in different tissues in the body. So it's not going to be enough to see what's going on in blood. We'll have to understand what's going on in gut. We'll have to understand what's going on in brain and potentially other tissues as well. So it's a very complicated picture, but again, science is knowledge. And what we learned in the last 15 years is very important. So let's start by saying mechanisms of persistence. This is going to be very simple. The general idea is that HIV can persist in immune cells in the latent form. It integrates in the host genome, and that it has a second particularity, is that if a virus integrates in a cell and that cell proliferates, the virus will proliferate with the cell. So that is a way to expand the reservoir that we did not understand previously, but it's going on, it's happening, and it's also a limitation in the different strategies that we do have. So as I said, HIV DNA into the host DNA, and we have to understand two things here. One question is that, is this virus intact or is defective? And I'll give you some clues on that. And the second point is that it's important where this virus, it integrates in the DNA. DNA is huge, and there are many codifying areas, areas that they do not express proteins. How important is the area where the DNA is integrated in? And I'll give you one example. And we've been working over the last 2 years in what we call extreme phenotypes of HIV progression. So most of the people in HIV will be what we call normal progressors here in the orange area. And that means they will develop, in absence of treatment, some viral load. And if you take this progression to disease as a CD4s, they will be losing CD4s progressively. But there are people that departure from this normality. And some people who get infected and progress very quickly. These are the rapid progressors that we did a study profoundly in the past. And there are also people who, in the absence of treatment again, they will control HIV. Low viremia, but high CD4s, low progression. And I did not want to go into this other phenotype, but it's very interesting. We have a person in the lab working on that are people who, despite having lots of virus and not treatment, they don't get sick. And they preserve their CD4s. And this is something that is not going to be presented today. But I want you to focus in this group here, the HIV controllers, in a very specific group we call exceptional elite controllers. Why exceptional elite controllers? Because these are people that have been without treatment, but infection for more than 25 years. Not treatment, again. And they don't have viremia, and they preserve their CD4 T cell counts. There are few people identified with this phenotype, and as I said, this is a little bit different from normal elite controllers. What I want to highlight here is that if you look and you ask, do these people have HIV DNA in their cells? The answer is yes, they do have it, very low levels, but they do have it. This in gray area would be the normal level of viral load, of proviral load, and this is what they have, detectable. But on the contrary, those viruses, they do not replicate, they do not express viral RNA, and they don't have viremia, plasma viremia. Even when you measure plasma viremia with very sensitive methods. So there are many reasons for that, and it's a review now coming up in Med on this phenotype, but I want to give you one idea from the viral point of view, is that if you get those people and you sequence the provirus that is integrated in the cells, one thing that has been across different studies, and this is one of our studies, is that despite many different amplifications, what we found, and these are 3 different people, that the virus is not complete, it's defective. So it's not going to be able to produce a new virion. Even in the only case, the only sequence we found that it was almost a full sequence, there was a mutation in the splicer donor site that it preclude that sequence to be producing a new virus. So one of the messages is that most of these people, they do have DNA, as you saw before, but this DNA is defective for the virus, so the virus will never be produced. And the second point I want to make here, and this is a study from my colleague Xu Yu in Harvard and Roentgen Institute, is that sometimes the virus, even being fully replication competent, it does integrate in areas of the genome that are not competent for replication. They are silent areas. The virus gets there, but it will not transcribe. Even being intact, it will not be able to produce new virions. So the 2 reasons: first, effective virus; second, integration in areas that are not ready for transcription. And there is a third point that we did a study in the group that is, well, you might have a virus in the area that is replication competent, but what happens in terms of epigenomic regulation? And epigenomic regulation is a complete new dimension. It's very complex. So what we did here is we studied different people without HIV, people with HIV but not treatment, and the same people on treatment afterwards. And we did study CD40 cells asking for the epigenomic trace in those people. And this is very complex, I know. Each of these numbers is one chromosome, one human chromosome, and this is the epigenomic profile across the different chromosomes in the body, comparing people with HIV with people on with HIV, people with HIV and the same people after treatment. You may ask, say, look, Javier, that is impossible to understand. This cartoon is very complex, I agree. Let me make it extremely easy, probably too much easy. So people without HIV, what happens if these people get infected? What happens is that automatically the DNA of those people gets hypermethylated. There is a significant increase in the methylation of the DNA of these people. And the question now remains is that what happens if we treat these people? Will methylation go back to normal or will not? And the answer is that it does not. So once the person gets infected, the DNA gets methylated, and some of these traces of methylation will never come back to normal. So infection, despite of treatment, will introduce modifications in the DNA that are hard to modify in the future. Second point is how to measure HIV reservoirs. This is it's been complex. I want to make only a few points here. One is that if you look at this replication cycle of HIV, we can measure the number of infected cells. This is what we call proviral DNA. We can measure the ability for this virus to transcribe, and this is in blue what we call RNA expression levels, or then we can look for residual plasma viremia, and this is done with techniques that are more sophisticated than the regular viral load that we use in the clinics. It can be much more sensitive. And finally, we can do assays to understand whether the virus are defective or replication competent virus. And this is what you have in orange. So by implementing all these studies, we can benefit in terms of clinical studies how to try to deal with the level of the reservoirs. This is a cartoon that we made in a review in Nature Medicine within one of our Martin Delaney collaboratories, and it tries to give some kind of clue how to explore viral reservoirs in the clinics by using blood or by using leukapheresis which, most of the times when you have very limited amount of reservoirs, a regular sample site is not enough. You need to get a lot of blood, or you can go to lymphoid tissues, either gall or lymph nodes, and how to do the different assays. One thing that I wanted to mention here, and this is what we did some time ago, is that when we went to the gut can be very rich in lymphoid tissue, as you know. And we wanted to look into ileum. And to do that, we developed a technique that will basically purify human leukocytes from the tissue, and will get rid of everything else by isolating the human lymphocyte with non-enzymatic techniques that will preserve the label markers of the cells. We can increase the level of detection. So this is, on your left-hand side, is what we will see without purification of the human lymphocyte from the lamina propria. And here, this is digital droplet PCR. So in green, you see the signals. Exactly the same sample if we process the sample by isolation of human leukocytes from the same tissue and the same patient. So very, very good. And in terms of clinical studies that we did afterwards, you see that in the lower side, you have what we detect in blood, and in the right-hand side, you have what we detect in ileum tissues, which is higher than the detection in blood. So there are tissues in gall that can be more sensitive to detect providers of HIV. And the other study that I wanted to bring here is that in the last 20 years, probably, there's been a large amount of different cell subtypes that have been proposed to be part of the viral reservoir. So what we did with Christina Galvez in the laboratory was to get a huge study on, a half a liter of blood from different donors and say, well, this is all the framework of what has been studied so far. Let's try to look all together, not individually, but all together within the same samples, what is the level of the reservoir. So we measured the reservoir, and what you have here in this right-hand side cartoon, there are 3 parameters here to look at. This is the total HIV DNA on the x-axis, but remember that depends on the volume of blood. You might have more of those reservoirs in the whole body or less, and then the size of the cell is proportional to the ability of that cell type to produce viral RNA. So what you can see here is that there are cells that are very effective in harboring HIV DNA, like CD32, follicular helper, CD20. But at the end of the day, the cells that are more present in the body with high reservoir would be cells like CD4 with a phenotype of transcriptional memory markers. So this has 2 messages. First is that the reservoir is not unique in one cell type. There are multiple cell types that will harbor the reservoir. But second is that there are some cell types that are more effective in not only harboring the reservoir, but producing new viruses. And this is, following on that study, we thought, what happens on the people we have on antiretroviral therapy? So when you have people without therapy, you have people with high viral loads, people with lower or medium viral loads. But when you have people on therapy, something very similar happens with the proviral DNA. So if you measure proviral DNA in 450 people, you have a whole repertoire of people with different reservoirs, from more than 1,000 to almost none. And what we did was to study the reservoir, and we pay a lot of attention in the people that we call low virate in the right-hand side here in blue, which are the people that have less than 50 copies of HIV DNA per million PBMCs. Some of them, they don't even have detectable HIV DNA. And I don't show that to you, but you can see that on the papers. These people, they do have also very small or undetectable reservoir. When you go to lymph node or you go to gall, it's very consistent. What we see in blood is very proportional to what we see in lymphoid tissues. So these are regular people. They've been on antiretroviral therapy for more than 3 years. They come from 2 different clinical sites. And the story is that why these people have such a low reservoir? How can we mimic that? One important thing on that cohort is that these are people where, when you look before therapy and after therapy, they also had a low proviral reservoir before therapy. but the decline is even much faster in the low-viral people than in regular people. So this is the decay of proviral DNA in most of the people, and this is the decay of proviral DNA in the low-viral reservoir in blue. So a 16-fold greater decay than regular people. We want to understand that in the group. We want to understand why that people have such a decay in the reservoir and how we can benefit on that. We are now part of one Martin Delaney collaboratory that is very much focused in this cohort of individuals. Regulation of latency, that's another topic that we are very interested in. You know that there are probably two types of trying to perturbate the proviral DNA in the cells. One is by the shock and kill strategies, that if a cell is latently infected, nor the treatments nor the immune system will be able to recognize the virus. But if we activate that virus to produce viral proteins, the immune system should be able to recognize that that cell is infected, and it will probably kill that infected cell because it's producing viral proteins. That is one possibility, and there are a number of different compounds that have been proposed to do that job. And the second is what we call the block and lock. Is, let's say, let's get the virus in a deep sleep, in a deep dream, and see whether we have any compound that can block definitively the DNA in the human DNA. And in that sense, in the group, we are very interested in how we can achieve, especially the second goal, by interventions in the transcription, the splicing, and the nuclear export of the viral RNA. So I know this is very molecular biology, but it's very interesting because the goal is basically, can we get rid of any transcription by understanding host factors in the cell? And we are screening multiple compounds from different libraries to understand whether we have a way to preclude, for instance, viral export from the nucleus to the cytoplasm. If we can do that, we can lock the virus within the nucleus and will avoid the production of viral proteins. I'm going to give you just a couple of examples. This is from a drug called ABX646, and it's from a French company, actually. And when we look into a clinical trial by Sylvia Bernal on this compound, only for weeks of treatment, they declined. These are individual people. There was a decline in the proviral DNA in basically all these people. And when we look more specifically into why that happened, we also detect that there were some defects in the transcription of the virus that were induced by the drug in two different steps. There are 5 steps in the transcription. So the 2 that have the red arrows, the initiation and the 5' elongation, we detect that the drug was able to perturbate these steps in the viral transcription process. So one of the ways we do that, and I before mentioned about different assays of measuring the reservoir, this is a novel assay that we're very happy with because it's been implemented now by the NIH and different other institutes in the world. It's called BeepSpot, and BeepSpot tries to fill a gap in the knowledge that we had and the assays we had to measure the viral reservoir. The BeepSpot is based in the early spot technology that some of you guys are doing with the CD8 T cells, and basically will tell us the cells, any individual cell. It's an assay that was cell-based, any individual cell that is able to produce viral proteins. Doesn't have to be a virus. It has to be, in this specific case, is a P24 protein. So when you get that, what you get are these spots in the plate, and the more spots, the more cells you have that are able to produce the virus. This is very important because this is what we measure, the total achieved DNA. Some of you have been here about the APDA assay, or the intact provirus assay, that it detects only the viruses that are full competent, and there are fewer of them. But when you look of those, how many of them they are able to produce viral proteins, there is a significant decay. So not all the viruses that are intact are still able to produce viral proteins. And this is what this assay is doing. This is an assay that is high throughput. So you can run a 96 well plates. It's ready for clinical assays. And this is why it's being widely implemented now in different studies. Immune therapies. A couple of words on immune therapies. I know there is now a talk on immune therapies, which will be more insightful. But let me give you some insights into that. We've been working with checkpoint blockers, specifically one of them, pembrolizumab, in people with melanoma. And what we can tell you is that we do see signatures of modification of the immune responses. This specific case is a patient with melanoma that was in complete remission thanks to the pembrolizumab. However, the viral reservoir of this person who was infected by HIV did not change over the course of the pembrolizumab intervention. And the second study that I want to show you is this study that we did with anti-PD-L1, durvalumab. And the goal in this study was to understand whether people with HIV, with different malignancies, they could benefit from receiving checkpoint blockers. You know that most of the people with HIV, they are excluded from the studies with with checkpoint blockers. And we want to do this study to just prove that also people on antiretroviral therapy, they could benefit from these studies. And the key point on this article is that even people with low CD4s, they would benefit for having checkpoint blockers, in this case durvalumab. And now we are analyzing the reservoir, but I can advance you that the changes that durvalumab induce in the viral reservoir are not very important, are moderate, let's say that. And also mention on vaccines. This is a vaccine study that was developed in our institute by Christian Brander and his group. And what they did is that they had an immunogen called HTI that they administered in form of DNA and also in different viral vectors. And what they wanted to do is to see whether the virus rebounded after treatment interruption or not. And one point that is important is that when they look at what is called the antigen immunodominance before the therapy and after the therapy, you know, the immune system is really re-educated to respond to the vaccine. And when they stop therapy, and this is the placebo group, and this is the vaccine group, what you see is that there is a small difference between the vaccine group, 31% controllers, versus the placebo group, 20% controllers. This is a non-significant difference, but there is some effect there that they are now trying to harness and see whether they can improve that. And finally, a few words in a study with broadly neutralizing antibodies. This is a study we did with colleagues in Denmark called ICLEA, and you know that antibodies are important to neutralize HIV, but antibodies have other different functions as well. And they do have non-neutralizing activities, which are mainly related to phagocytosis, macrophages, NKs, and cell death. And the point in this study was to try to understand whether using broadly neutralizing antibodies, we can reduce the reservoir. And what it was done in this specific study was to treat people early after infection. And within 1 week of treatment, when you still had detectable virus, the antibodies were infused. And the idea was to understand whether the immune complexes that are formed from HIV and the antibodies could lead to a degree of control, some degree of control to HIV replication. What you can see here in this plot is that in red you have the people who had antibodies to which the virus were resistant, but in blue you had the people with antibodies that could neutralize the virus. And what you can see here is that there is some level of control after treatment interruption on the people who were on antibodies with sensitive virus to those antibodies. So they can do something. And finally, I want to give you some clues in terms of cell and gene therapies. I'm very appealed with this topic because we've been working a lot with in the last 10 years, and people here at Pasteur with us here have been also part of this study. Let me tell you one story. I'm traveling back like 30 years. So in the 90s, there was this this headline in the newspapers, in the American newspapers. It said, the man who couldn't catch AIDS. It was called Stephen Cronin. He was a public person, a person who was exposed to the virus multiple occasions and had friends who passed away because of HIV, and he couldn't get infected. He never got infected. And we understand at that point that the reason was that of the two receptors that the virus needs to infect the cells, CD4 and CCR5, Stephen Krohn did not have CCR5. And he did not have CCR5 because he had a mutation, a deletion of 32 nucleotides in the CCR5 gene that preclude in homozygosis to produce this protein. Therefore, the virus could not enter the cells. It took about 20 years to make this observation something that we could use in the clinic. And in 2009, Timothy Brown, called the Berlin patient, he was a person with HIV who had leukemia, and he needed an allogeneic stem cell transplant. And his hematologist, Hero Hutter, in Berlin, he thought, well, if we find a donor that not only was compatible with him, but he also had the same mutation that Stephen Krohn had, the deletion of 32 nucleotides in the 2 alleles in CCR5. So he found the donor, and Timothy Brown was transplanted, and he's been able to live for 13 years with no leukemia, no HIV. So he is the first person reported to be cured by a medical intervention. But that happened in 2009, and ever since we couldn't repeat that experiment. It was very complex. We couldn't understand whether that was a fluke or not. It was very disturbing. With that idea, we enter in this world, and in 2014, an hematologist from another hospital in Barcelona, they called us, said, look, we have a person here. He is HIV positive and he needs a transplant, but he's a journalist. He knows a lot, and he knows about the Berlin case, and he wants to become the second cured person in the world. So with that idea, we started working with our hematologist fellows in Barcelona, and we found a donor for him. Everything went fine. He got the transplant. In that case, was from cord blood cells. It was not an adult donor, but everything seems to be fine. The transplant was fine, but the problem was that a few months later, that person passed away from his, it was a lymphoma in this case, and he passed away because of lymphoma. However, all the follow-up we did on this person, his cells were completely reconstituted. They did not have CCR5, and potentially he was one person to be ready to be cured. That study brought us into a consortium called ITHISTEM. And ITHISTEM started working in 2014. I'm co-chair with Anne-Marie Wensing in Utrecht Medical Center. And ITHISTEM was an observational project. It was not a clinical trial to investigate cases of allogeneic stem cell transplantation in people with HIV. If you want, very few cases. We had different centers in Europe, including Paris, and we had other clinical associated sites in many different countries in Europe, and one in Canada as well. And we did that mainly in Europe because the prevalence of CCR5 delta 32 is greater in Europe and is more prevalent North Europe than South Europe. And one of the things we did in the study was to genotype almost 5 million donors to understand whether they had the CCR5 or not. And this is now reported. I mean, if you go to the databases, or your hematologists go to the databases, this is downloaded there, and this can be found in the hematologist databases. And with that information, we defined the second case of cure, which is the London patient, Adam Castillejo, who is now 6 years cured. We published that in 2019 in Nature. I have to say that I'm always proud of publishing in good journals, but in that specific case, I'm even more proud that we got a tweet from Donald Trump saying that he was very happy that the second person had been cured of HIV. I don't know what's the impact factor of Donald Trump tweets, but as far as I know, I think they're high. By the way, there is an index. You know, the impact factor, there is the Kardashian index that follows on the ability of scientists to be public in the media. So you can follow that, Kardashian index. And then 2023, Mark Franke, the Düsseldorf patient, was also cured last year. And he's been 4.5 years of treatment, and he became the third person cured on HIV. And after that, we had 2 more cases. The City of Hope patient, and the New York patient with a little bit shorter follow-up, but still also cured, and there are more cases to come, and hopefully in the near future. This is information not from a specific people, but also from all the people in the cohort. They're finishing. More than 30 people in this cohort. And what I want to show you here is that this is the level of reservoir before the transplant. People on treatment, they had this reservoir level. This is what happened after achieving full donor chimerism. It's 2 logs decrease. Remember, this is log scale, so very significant. And you might wonder whether this is dependent on doing the transplant with CCR5 delta 32 or not, and the answer is not. So the decrease, this is decrease. So this is 2 log decrease. The decrease is the same whether you do the transplant with CCR5 or not. So there is a donor allogenic immunity, what we call the graft versus host or graft versus reservoir effect. So for reducing the reservoir, the delta 32 is not important. The delta 32 is important to avoid any potential rebound of the virus afterwards. And with the 30 people, we did some modeling in collaboration with Fabian Cardoso in Seattle. And what you can see here in this cartoon on the right-hand side is that this is total HIV reservoir. In black, you see the effect of the transplant in people who have graft versus host effect. If that person does not have a graft versus host effect, what you get is the red line. So no decrease in the reservoir. And this is modeled across tons of data that we had in this study. So you can tell me, and you are completely right, Javier, this is not feasible, this is not scalable, we cannot transplant people with HIV, and you are completely fine on that. But we're trying new things, and in the lab and other labs are working on gene therapy, try to see what populations we can harness to deplete in vitro by using CRISPR and other techniques, including TALENs, the CCR5, and do autologous stem cell transplantation, which is much safer and can lead to the same result. Want to end by reminding you 3 things. First is that in 40 years of HIV, I think the community and the medical community and the stakeholders have done a great job in developing and implementing new prevention techniques and new prevention strategies. Treatment for HIV has been clue, and we know that undetectable equals untransmissible, and we have great drugs now that can control HIV. But we still have to cure HIV. There are many people out there that wants to be cured, and we need to find the ways to cure HIV. And I want to say, not only for HIV, we might have a pandemic in the future that is also a provirus, a virus that can get integrated in DNA. Any research we do to understand HIV and how to get rid of the provirus can be useful for other pandemics. And science is going to be clue in that work. And this, by implementing prevention, treatment, and cure, that's the only way to get rid of HIV in the planet. I want to thank all the people that participate in the studies, and there's a lot of generosity in these people, and I will profoundly acknowledge them for all their commitment. And also want to thank the people, the facility, and all the sponsors that we have for all these studies. And thank you all for your attention. Thank you very much.